This is chapter 26, objective 5, the regulation of acid-base balance. This is the last objective for this chapter. Now, we're not going to get into a lot of the chemical equations that we could for this, so we're just going to kind of do more of an overview of the regulation of acids and bases instead of getting into the intense chemistry aspect of it. So, hydrogen ions affect the activity of enzymes, so they can denature them. Hopefully, you all remember about the pH chapter, where enzymes are extremely sensitive, and if you get them in a high acid environment, they're just going to break apart and denature. And keep in mind, once a protein denatures, it can't fix itself. You know, it's gone forever. It has to be remade. They can also interact with negatively charged particles. Now, chemical reactions are highly sensitive to those hydrogen ions. The lower the pH, understand the relationship here between the pH and the hydrogen ion concentration. So the lower the pH, so like a pH of 1, 2, 3, is going to have more hydrogen ions, meaning it's going to be very acidic. There are 3 mechanisms that help regulate the hydrogen ion concentration. You've got buffers, you've got the respiratory system, and you've got the kidneys. So buffers instantaneously resist a change in pH of body fluids. Buffers bind to excess hydrogen ions, or they release hydrogen ions depending on what the body needs. 3 important buffer systems are the carbonic acid bicarbonate buffer system, the protein buffer system, which would be hemoglobin and plasma proteins, and the phosphate buffer system. So just showing you a little bit, I'm not going to ask you this, I'm just kind of showing you what's going on here. Carbonic acid, H2CO3, is considered a weak acid. Take a look at these arrows. See how they're going opposite each other? That means this reaction goes forwards and backwards. So carbonic acid turns into hydrogen ions, and then the hydrogen ions can be combined with the bicarbonate ion and then form the carbonic acid. So this is constantly flipping back and forth. When hydrogen ions are added to solution, then carbonic acid forms. So when you've got hydrogen ions in solution, it will reverse and form carbonic acid, lessening the amount of this free hydrogen, making the area less acidic. When hydrogen ions are removed from solution due to the presence of a strong base, bicarbonate ions would be considered a strong base, then the carbonic acid adds more hydrogen ions to solution. Now, the protein buffer system, hemoglobin in red blood cells is one of the most important intracellular proteins. This is a protein within the cell. We know that it binds oxygen, but it's also a protein, and that can act as a buffer. They have certain chemical groups called functional groups that are attached to it that can either be considered a weak acid or a weak base. So think about this large protein structure, and then it can have little groups of atoms coming off of various parts of it, which are called functional groups. And sometimes those little groups of atoms coming off of that protein molecule could either act as a weak acid or a weak base. If you have a weak base as a protein, then when hydrogen ions are present, the hydrogen will bind to them, lessening the hydrogen ion concentration in the blood. If you have a weak acid as a protein, then hydrogen ions can be released, increasing the hydrogen ion concentration. So know the relationship between the weak acid and hydrogen ions and what's happening. Or is it becoming more acidic with a weak base and hydrogen ions, or is it becoming less acidic? That kind of information. So here I'm just showing you, you don't have to know the structures, I'm just showing you what's going on here. So we've got a weak base, we've got this amino group right here, right here, H2N, that N right there lets you know you've got an amino group right here. The weak acid, we've got a carboxylic acid group. So we've got this amine group coming off up here as well. This is part of the amino acid, but you have this extra amine group coming off the top. And then you also have weak acid. So yeah, you got a carboxylic acid right here off that main alpha carbon, but you've also got that carboxylic acid group right here, which makes it acidic. And this up here makes this a little basic. So those are those little functional groups that are coming off of proteins. Now you also have the phosphate buffer system. DNA, RNA, ATP, and phosphate ions act as buffers. So you should recognize these 4 compounds. When the pH decreases, That means you're going to have more hydrogen ions around. Then hydrogen will bind to the biphosphate molecule. So this right here is biphosphate, and this is hydrogen. So when you got a lot of hydrogens around, these two will bind together. This is a double arrow. When these two bind together, the reaction proceeds in this direction. When the pH increases, fewer hydrogen ions, meaning it's more basic, then the hydrogen will be released from the dihydrogen phosphate. So this right here is dihydrogen phosphate, and the reaction will go this direction when you're running short on hydrogen ions. So again, this is that buffer that allows you to either take up those hydrogen ions if there's too many, or produce hydrogen ions if there's not enough, to try to make sure that balance is of pH is that 7.35 to 7.45. And that concludes chapter 26.